Hi everybody. I wanted to make a real quick video to give you a very, very fast rundown introduction for some possible project ideas. This is not meant to go into a lot of detail, and I'm not going to edit it a lot. I just want to run through some ideas real quickly to give you some things to start thinking about for what your projects might be. If you want more details on any of these things, I'm happy to go into to more details with you later on. I want to give you just a sort of a brief bit of structure to this, and it's not perfect. I want to try to break down these project ideas into projects that are based on particular organisms or group of interacting organisms, projects that are focused on particular kinds of systems or habitats or a particular kind of interaction, and then finally question-based projects where you start out with a particular hypothesis or a research question already, and then you're just going to look around for a system afterwards. This hierarchy here is not at all perfect, and you'll see how messy it is when we start talking about this through the semester. But this is just to kind of organize my thoughts, and we'll come back to this in a few other discussions that we have throughout the semester. So first, organism-based projects where you focus on one particular species first, and then you kind of try to learn more about it afterwards. So for many of you, this won't be a surprise. I'm really interested in moths, and I've done a bunch of moth-focused work. Moths and butterflies are easy to work with. They're abundant. They're all over the place. And I am going to highlight a couple different kinds of questions that you can look at throughout this whole presentation. But they're easy to sample for looking at different kinds of substrates or surfaces. You can attract them with some equipment that's real cheap and easy to work with, and that I can send to you if you're not on campus. You can also set up a sheet or have a bright light and just go and look for them at night. So that would be focusing specifically on moths. You can also focus specifically on mosquitoes. I've got some mosquito collecting equipment. You could look at questions like how many of them are there, how many different kinds of species are there, so that's a community level question. We might be able to pull off looking at the abundance of West Nile virus, or I've got to screw up on this third bullet point here. There's West Nile virus, the incidence of West Nile virus in mosquitoes. That requires some lab work. Or maybe insecticide resistance of mosquitoes. So that's I conflated two different things in that third bullet point. Like I said, I'm not going to edit. Plants are real easy to work with. So doing a plant survey either at a particular site like this Ruffner Quarry that you see here. This is a project that a student of mine did 8-9 years ago, where she just focused on plants that occurred in that one habitat. There are apps to help you identify these plants. You do not need to have gone through botany to be able to identify things. I can help you with the identifications. You'd either focus on a particular site or maybe compare plants that occur in different sites. There are a whole bunch of projects out there that are citizen science-based projects. I'm going to give you examples of just a couple in this video. One of them is called Project Squirrel, which is an awesome name. They do projects where you can do some very simple experiments. Basically, you're feeding squirrels and watching their observations and collecting data on how much they feed in safe places versus unsafe places, and with safe being sort of away from or near trees, and unsafe places being farther away from trees. So citizen science projects are nice in that you're basically helping other people collect data for their larger projects. And so there are some protocols set up. I've done this with past field ecology courses for sort of individual small labs, but if someone really wanted to dig into this, there's a lot of possibilities there. Next is going to be system-based projects where you're looking at a habitat or a group of habitats. You can probably already see how my breakdown of question-based and system-based and species-based or organism-based is already kind of fallen apart here because I've talked about particular kinds of habitats already, but we'll ignore that. So I'm really into pollination, and pollination is not exactly the same thing as flower visitation. There could be something where insects are robbing plants, so they're taking the nectar but they're not actually pollinating. So here's a few images of that. So you could potentially look at either pollination or visitation. The species that are visiting particular flowers are of interest either in the daytime or nocturnally. This is a picture of a moth that is visiting a particular plant. Now that plant is of interest to me. This is a study that I'm working on right now. But you can see that proboscis is out of that moth and it's actually taking in nectar and probably pollinating the plant. So if you were to follow a particular plant for a series of sampling periods and just collect data on what things come there, you could net those individuals, again, using some phone apps like with Plant ID. You can use some phone apps that can really help you identify these things, and I can help you identify these things. So just following a particular plant, particular group of flowers, you could determine something about what species visit. And you would not believe how many species this has not been done for. So for this particular plant, nobody knows who pollinates that plant. So that's a real straightforward study. You could also kind of address the system of soil arthropods. So these are a bunch of very small insects or insect-like things that occur in the soil. You can use these things called Berlese funnels and sample one habitat, multiple habitats. What someone has done in this past project is these students have gone out and compared a whole bunch of different sites. You don't have to do a comparative approach, but they looked at just the total number of kinds of things, so different sorts of species that occur in these different places. And that the thing on the y-axis there is, or sorry, the thing on the x-axis for this particular figure is just species richness. You can also make this a little more of a question-based technique. You can look at along something called an ecotone. So an ecotone is a transition between one kind of habitat and another kind of habitat. So they were very interested in this concept of ecotones. They looked at some ecotones and compared ecotones along at two different habitats, Turkey Creek and out at the BSC Ecoscape. This is from another citizen science project or citizen science network called the LeafPak Network. So if you have access to a stream, you look at that upper right-hand picture, you can see a leaf pack. That leaf pack could either be naturally occurring or you could assemble one, and then you look to see what kinds of water invertebrates or aquatic invertebrates are found in that leaf pack. And again, you're helping someone by collecting data for them. And you can also design your own experiment that goes along with that. Another technique which is a little more system-based than others is using basically modeling clay to make a whole bunch of little caterpillar models that are then stuck out in a bunch of different habitats. You can either make them out of different colors or have different shapes, make them more conspicuous or less conspicuous, and then you can see what things tried to eat them. And so what students have done in the past is glue different shapes and different colors of these things on soils on different surfaces, and you can register the indentations on the surface of the clay to see whether birds have tried to eat them or ants have tried to eat them or maybe parasitoid wasps or mammals, etc. So that's a possibility. Finally, there's some question-based projects. So these are motivated by a particular hypothesis. So let's say you're really interested in the impacts of urban heat islands, and you wanted to investigate the approach of an urban heat island, or you wanted to see whether a particular modification to a habitat led to a particular kind of outcome. So those are hypothesis-driven research questions. Ultimately, you're all going to have to frame what your research question is, even if it's how many moths are there at my backyard, or how many different mosquito species are in these two different kinds of habitats. So you'll all have to come up with some kind of research question, but this last batch of project ideas tends to be more question focused, and then later on you try to find out what kind of system can you answer that question in. So again, a past student has worked on this question of, are there more parasitoids, these little enemies that you've probably heard about in evolutionary ecology? Are there more of these little insect enemies attacking herbivorous caterpillars in an urban setting or a rural setting? And so they actually gathered a whole bunch of caterpillars from 2 different sites and then fed them food until parasitoids emerged, and then they looked at different parasitism rates of these caterpillars. I had another student in the past look at herbicide resistance, the evolution of herbicide resistance in a plant species, and I've had students work on 2 different kinds of plant species. This is like many of these projects, you're going to have to get an early start on them, but you could look at herbicide resistance by comparing plants in different habitats, or look specifically for the evolution of herbicide resistance. I have a lot of interest in what moths eat as adults, not as caterpillars. A lot of that is already known, but what do they eat as adults? And in general, that's not really known very well for moth species. And like I said, moths are all over the place, so this can be a straightforward question to try to address. So I've got a couple questions. What do moths eat as adults? And then secondly, can their proboscis morphology, I'll talk about what that means in a second, or their gut bacteria be linked to their diet? These last 2 questions require a little more technical equipment to be able to address, but I'll mention that briefly. So proboscis morphology is the shape and the structure of the proboscis. So the proboscis is the mouth part, the feeding part of a moth, a butterfly or a moth. And what you see in this top image here is the proboscis straightened, and that is a SEM, or scanning electron micrograph image of that straightened proboscis. And you see the tip that's circled in red there. Those are some structural features known as Sensilla styloconica. That's why it's SS. And so the question that I've been trying to address with a colleague in Ohio is, if we take a whole bunch of these images of a bunch of different species of moths, do their proboscis structures or lengths or the presence of Sensilla styloconica differ depending on what the moths eat, if they're flower visitors or if they're maybe fruit visitors. Moths are also potentially pollinators, and I mentioned that earlier on. You could just follow a particular plant to see if moths are visitors to those flowers. But in general, all the other kinds of things that they feed on is relatively understudied. So they could be visiting particular plants as pollinators, but they could also be visiting lots of other kinds of things. So they could be attracted to fruit. They could be attracted to carrion or dead animals. They could be attracted to animal dung or a variety of other different kinds of food sources. So what we've done in the past is we've taken these traps, which are real easy to set up, and baited them. So you see in the yellow box around the top figure there in the middle, we've baited those traps with flowers. And then in the morning, these moths have come in and they can't escape. So presumably anything that came into the trap during the evening was attracted to flowers. They're probably flower visitors. And the image below with the red and orange box around it, we've baited those traps with fermenting fruit, and a whole different suite of species will come to that fermenting fruit. So if you live in a place where you could hang up one of these traps, you could bait it with a variety of different things to see what sort of moth species come to them. And then another area that I've been working in quite a bit with students recently has been to address questions of their gut bacteria. Now, my original plan for this summer was to follow up on all these different kinds of questions that students addressed last summer. Unfortunately, we're having to meet remotely and not in person, and so a lot of these things are very lab focused and lab intensive, and so I'm not sure how many of them are actually going to be possible. But I'm going to talk about it real briefly anyway. You may want to pause on this if you are interested in any of these questions, and then read through them. But I'm going to charge on. One of the things that we've done is we've taken samples from flowers, fermenting fruit, or maybe just soil, or any other things that we're seeing out on the trail. And we can take these samples and we can process them and send them off to a lab at UAB, and they can tell us what kinds of bacterial communities are there. And this is a dizzying display of a bunch of different colors. Each color shows you a different bacterial species. So for example, if we look on the far left, sample number 42, that was taken from the digestive system of a butterfly on some dog poop, and almost all the bacteria in the gut of that butterfly was 1 or 2 species of bacteria. But if you look maybe way over on the right, sample 78, if you look at a sample of mud, you see a whole bunch of different species of bacteria. So we've been looking at a variety of different food substrates and gut contents of moths and butterflies to see what their bacterial communities look like. And one of the questions that we've had is, hey, if you look at a bunch of different food sources, do their bacterial communities kind of seem similar? And it sort of looks like it. You can see that I've drawn some ellipses around samples that came from dung, samples that came from flowers, and samples that came from fruit. And yeah, they sort of all are somewhat similar in the kinds of bacteria that they have in them. So that is a possibility for students to work on. It does require some specialized equipment, which I'll talk about in a little bit, and potentially access to BSc, but not necessarily so. And then I want to end with a few considerations. So in order to be able to do a field research project, you have to have access to the field. Everything that I've talked about is related to you going out in the field and conducting a study. Ideally, that's what we're shooting for. If none of these are possible, if you don't have access to particular sites or habitats or even a front yard, we can come up with something. We'll be able to work something out, but you got to let me know that that's your situation. Maybe you've got a lot of different habitats and you could choose from a variety of different questions that you're interested in. You might need access to some specialized equipment like pipettes or DNA sampling equipment, or those traps that I showed you that we could bait with different kinds of food. Some of these things I can send to you. Some of these things you can borrow for the summer. Some of these things, if you don't have access to it, you can't do this study, and we don't have access, so you can't do the study. So we have to consider that in choosing your question and your study system. It would make it easier if you could come to campus occasionally, but not everybody is living in town or has easy access to campus, and I'm not sure at this point whether students are actually allowed onto campus to be able to do research. So I would have to check into that. But if you did have access to campus, that might make some of these experiments and projects a little more achievable. The timing is also another issue. If you want to set up an actual experiment where you manipulate something and make it grow and then come back and monitor it or whatever, that takes a lot of time. So we got to set those up soon. If you want to follow the pollination of a particular flower, it's got to be flowering during the time that you're doing the study. So timing is a real issue. We don't want to get involved in studies that are going to take 8 weeks to do, because that's going to take you 8 weeks on top of all the other things that go along with this. And then finally, we have to be very concerned with keeping everybody safe. When you get access to a field site, if you're going out on your own, there might be a potential risk. Sometimes doing work outside involves some other risks, like snakes or falling down or something like that. So we'll also have to take into consideration your safety in the study that you do. So this is a real quick rundown. This is not by any means meant to be a limitation on the things you can and can't do. It's just meant to give you some ideas to start out with, so that we can start having a conversation about the sorts of things, or maybe the approaches that you're interested in. I hope you'll ask me some follow-up questions either over your email or over Microsoft Teams. Talk to you later.